Hello, I'm Riya Gupta, and today I'm here to tell you about a gene signature for autoimmune and infectious diseases created using gene expression data from all around the world. When patients with an underlying autoimmune condition report life-threatening symptoms, physicians need to quickly determine whether these symptoms are caused by an acute infection or a complication of their autoimmune condition. If someone is suffering from a complication of their autoimmune condition, giving them drugs to treat an infection would not treat the problem and would allow the patient's condition to worsen. Immunosuppressive drugs that are used to treat autoimmune diseases can be harmful to someone with an infectious disease because their immune system would not be able to fight off the infection. So, an accurate diagnosis is critical to patient survival. Our goal was to formulate a gene signature to separate infectious, autoimmune, and healthy samples based solely on gene expression levels in the blood to help with quick, accurate, and non-invasive diagnosis. We would start with the patient and then use an autoimmune and infectious versus healthy signature to separate the healthy and disease samples. Then we would run this through the autoimmune versus infectious signature, so we know which general category of disease the patient is experiencing. From here, we can run more tests to get a more specific diagnosis. By first running the initial, overarching, and more general signatures, we are able to improve the performance of existing diagnostic methods. Here is a breakdown of the data we used, all of which is publicly available. Historically, a lot of biomedical studies have been done on a very homologous population, which has contributed to the reproducibility crisis because the work often can't be translated to new populations. Our gene signature is built from 122 datasets, comprising of 14,587 patients with 7,534 genes. This data has heterogeneity across country of origin, specific disease, age, sex, ethnicity, and many more factors. This is a really good thing because all of this diversity will allow the signature to be universal and much more applicable to any population. We can see that there is also a lot of heterogeneity within the autoimmune and infectious disease categories. This allows the signature to pick up on overarching patterns and be more generalizable across all autoimmune or infectious diseases. Since we use such a wide array of data from so many different datasets, we need to get rid of any batch effects that are caused by the technical differences between the datasets. So we coconut co-normalize them to make all the datasets comparable. Coconut takes the healthy samples from each dataset and determines a set of statistical adjustments that allow them to be comparable. Then it takes these adjustments and applies them to the diseased parts of the datasets. This allows us to combine the datasets while maintaining the distance between healthy and disease. In this image, we can see the top PCA graph is clustered based on datasets, and the red and blue samples are not clearly separable. This clearly indicates that there are major technical batch effects in the data, and it is impossible to run any useful statistical analysis on data that looks like this. However, in the bottom graph, which is represented by post-coconut co-normalization, we can see how you can no longer see each of the individual data sets. Rather, you can identify the patterns in the data from the biological differences. The data is split into three groups, with 20% of the data, or 27 datasets, being put in independent validation, and the other 80%, or 95 datasets, for training. The training data was again split, with 80% of it being used for discovery and 20% for holdout validation. We used a statistical multi-cohort framework called Manatee to create our signature. It uses differential expression statistics to first filter out genes, and then it creates a concise signature by adding or removing genes that increase performance in the discovery data. Then we finalize the signature and the parameters used to create it in the holdout validation data. Then this signature is run on completely new data as independent validation, and this is the best representation of the real-world performance. The first signature we found was one to separate autoimmune and infectious diseases from healthy samples, and it has 39 genes and achieves an AUC of 0.91. An AUC is the measurement of the area under a ROC curve, and it is used as a metric for diagnostic tools. An AUC of 0.5 means the model is just randomly guessing, while an AUC of 1 means the model is perfect. An AUC of 0.7 is considered to be good, while one of over 0.8 is considered to be excellent. And for this signature, our AUC was 0.91. We can see in the B-swarm plot on the right how the samples can be visually separated, as we can see how the red or healthy samples are below the dotted line, while the blue samples are above the line. In these forest plots, we can see specific genes that are up and down regulated between our samples in the independent validation. On the x-axis, you can see the effect size of all the genes listed on the y-axis. The blue box shows the effect size, while the line represents the uncertainty for each gene. The uncertainty is pretty low, which means that the signature is consistent. We can see how these genes have an effect size with a large magnitude. One thing we did was dig into the biology of the genes found in the signature. We can see that the signatures hold biological plausibility because there are several genes that are known to be associated with the immune system and autoimmune and infectious diseases. There is one gene here in particular, IFITM1, for which we can see the forest plots on the bottom of the slide. We can see how the effect size of IFITM1 varies greatly between the healthy and autoimmune and infectious samples. IFITM1 is known to work closely with the innate immune system, and the IFITM proteins are interferon response proteins and are known to be upregulated in response to infection. The second signature we found separates autoimmune from infectious samples, and this one has 22 genes and achieves an AUC of 0.88. We can see in the B-swarm plot on the right how the samples can be visually separated, as we can see the red or infectious samples are below the dotted line, while the blue or autoimmune samples are above the line. Here, we can see the forest plots for the genes in the signature, and which ones are up and down regulated, and by how much relative to each other. We know that the signature also holds biological plausibility because it too has several genes that are known to be associated with the immune system and autoimmune diseases or infection. We can see the forest plot here for one gene in particular, AKT1, and how it is different between infectious and autoimmune samples. AKT1 is a protein kinase associated with the mTOR pathway, and is very well studied and linked to critical cellular functions. So it is no surprise that such an important gene is involved in the mechanisms of disease. These signatures are able to accurately differentiate between these three groups and perform consistently across completely new data. There is a lot of heterogeneity in our data, which the model performs well with, meaning that it can be used across a diverse population in a more accurate, inexpensive, and non-invasive manner compared to existing diagnostic approaches. After these signatures are applied, additional testing is needed for a more specific diagnosis, and the initial testing helps improve these final results. One place we could use it is on a previous TB signature. Latent TB is when, although a patient is still infected, they don't experience symptoms, actively have the disease, or have the ability to infect others. Active TB, on the other hand, is when a patient is infected, experiences symptoms, and can spread the disease. Someone with latent TB can progress to active TB soon after getting infected, or at any other time when their immune system is weaker. The signature differentiates between people with an active TB and those with healthy latent TB and other diseases. It also tries to determine whether someone with latent TB will progress to active TB. We had a hypothesis that since this previous signature is trying to do so much, it could benefit from a triage test due to the less complex task of separating infectious samples from healthy and latent TB samples. This would allow the TB signature to be used on a population with a higher active TB prevalence. Then the two signatures could achieve a higher performance together. So this is the original 9-gene active TB signature. We can see the ROC curve here and some of the statistics. It currently has a sensitivity or true positive rate of 90.1% and a specificity or true negative rate of 81%. We decided to use the manatee analysis for the infectious versus healthy signature, but we needed to change it slightly to fit this situation. We used a very similar discovery strategy for this signature as the previous 39 and 22 gene signatures. The previous signatures were made to maximize the AUC, but for this venture, we wanted to maximize the sensitivity while maintaining a good specificity. That way, we would make sure to identify a lot of the people who are truly healthy or have latent TB, while minimizing how many active TB patients we lose. So, we took all the genes that Manatee chose with an effect size of over 0.8, and then ran a forward search on the discovery data from the TB project, so that we could get a better idea of how this signature would apply to that population. This found a subset of them that would be specifically useful for TB and work better for this scenario. These are the rock plots for these two signatures. We can see that the curves look pretty good and have mostly high AUCs. However, for the comparison of ATB versus other diseases, the infectious disease signature doesn't do as well, but the other diseases will be sorted out with the second signature. And for the first signature, it is more important that it does well when compared to healthy and latent TB, so we can identify those patients and rule them out for additional testing. To implement this method, our 17 gene signature would first be applied to the patients. If the patients tested negative, then they would not be further tested. However, if the patients tested positive for the first test, then the ATB versus other signature would be applied. Those who tested positive would be classified as having active TB, while those who tested negative would be classified as not having active TB. This way, it would have better results as a signature would be run on a population with a higher initial prevalence of active TB. We simulated the target population by assuming that 2% of it has active TB, 58% of it is healthy, 30% has latent TB, and 10% has another disease. This is more of an arbitrary number for simulation purposes. This way, we could calculate what category someone falls into, as well as what test they would get, and then see how the signature performs. Each box represents one category of the population. The green represents someone who receives a positive test, while the purple represents someone who receives a negative test. At the top, we can see how the original signature works. The bottom is a new approach. We found that the best combination was with the first signature at a sensitivity of around 97.5, and the second one with a sensitivity of around 92.5. So first, the patients undergo the new infectious versus healthy signature. We can see their test results here based on the performance of the signature on each of these categories at a sensitivity of 97.5. Those that test negative are immediately classified as negative. However, those who test positive are tested again. Using the sensitivity and specificity of the ATB versus other signature, we simulate the results of this test. Then we can see here how we combined the test results by taking the negative results from the first test and combining those with all the results from the second test to get the final tests for the entire population. Then we can use the final true positive and true negative percentages to get the sensitivity and specificity. We can see how after combining the two signatures, the performance is much better. The specificity went up by 8.13 without the sensitivity lowering. Diagnostic odds is a measure of how effective a test is. A 1 means that the test is just randomly guessing. While the higher the number goes, the more effective it is. The diagnostic odds ratio almost doubled. And this way of combining signatures can be used in the future and is a promising way of increasing the effectiveness of signatures besides better gene selection. I would like to acknowledge Aditya Rao, Lara Murphy-Jones, and Pravesh Khattri for their assistance and guidance on this project. I would also like to thank Khattri Labs and everyone in the Healthy Immune Response Project in particular for helping me with this project. Thank you so much.